Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and STEMCELL Technologies webinar titled Applications of Human Intestinal Organoid Cultures in Drug Discovery. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be the host. Today we're being joined by Dr. Victoria Conlin and Danny Leung, who will discuss the use of human intestinal organoid cultures in drug discovery and toxicity testing. And with that, I'm very pleased to welcome the first speaker, Dr. Victoria Conlin. Victoria, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks, Liam. Hello, and welcome to this webinar by STEMCELL Technologies, co-hosted with Scientist.com on the applications of human intestinal organoid cultures in drug discovery. I'll be one of your presenters today. I'm standing in for Johnny. My name is Victoria Conlin, and I'm the Senior Manager of STEMCELL's Contract Assay Services division. I'm joined here today by one of my assay development scientists, Danny Leung, and together we are going to talk about our new services that launched last year. But first, a little background into the biology of organoids themselves. Whether you're new to the field of organoids or have been working with them for a while, I want to recognize that these three-dimensional tissue models were given center stage back in 2017 when they were chosen by Nature Methods as the method of the year. This milestone highlighted the excitement in the field of biology towards the potential benefit of the inclusion of these models into basic research, and we have been working diligently to standardize the culture of these organoids in vitro from a variety of tissues for several years. The focus of this presentation will be human intestinal organoids. However, before I dive more deeply into the biology, I want to send out a quick poll to get some feedback on what kind of cells you are currently using in your research. I'll give you a second before I continue. So, as I said, our focus today is on human intestinal organoids, which are a highly versatile research tool introduced by Sato et al. In 2009. On the left-hand side of this slide, we show a schematic of the crypt-villus axis, complete with all the cells present in an in vivo setting. At the base of the crypt, you have the LGR5 positive stem cell niche, together with Paneth cells. Just above this niche, you have the transit amplifying cells in blue here, followed by the enterocytes, intermixed with a variety of secretory cell types, such as enteroendocrine cells and goblet cells, all of which form a polarized epithelial monolayer capable of absorption and secretion, which coalesce together in vitro to form a functional lumen. In the top right-hand side, we have an image of what mouse small intestinal organoids look like under a microscope, and you can see the branching sections of the crypt and the villus and the lumen located in the middle. These organoids can be maintained in stem cell media, expanded and cryopreserved, and either kept in a proliferative state or terminally differentiated into all relevant cell types listed in the bottom right of this slide. This slide shows how the proliferative and differentiated state can be achieved using our media. Starting from the left-hand side, we have intestinal organoid cultures in their expansion phase, which are grown in our intestinal organoid growth medium. From the immunocytochemistry staining underneath, you can see the proliferative cells labeled in green by the proliferation marker Ki67 and the epithelial layer outlined by the adherens junction protein, E-cadherin, seen here in red, and the nuclear stain DAPI. This proliferative state is often used by researchers interested in stem cell biology, cell proliferation, and tumor biology. Moving into the center images, we have organoids grown in our intestinal differentiation medium, and you can see the morphological change from a more cystic phenotype in the growth medium to a budding architecture in the phase contrast image of the differentiation medium cultures. Here, we have used the goblet cell marker MUC2 to highlight the switch from proliferative cell population to a terminally differentiated state. And finally, on the right-hand side, we highlight the utility of these organoids by taking them from a 3D to a 2D state and growing them as air liquid phase intercultures on transwell filters, where you can see from the ICC image the polarized nature of the epithelial barrier and the secretion of mucus on the apical side of these cultures, as seen in green by the MUC2 staining. Of particular relevance to our data being presented today is our human intestinal media versions, which enable the same proliferative and terminally differentiated states. The human intestinal organoids used in our contract assay services can be expanded and differentiated to fit the needs of our clients and the size of their intended assays. The top left ICC image shows the organoids in the proliferative state with high percentages of yellow Ki67 positive cells and a corresponding absence of goblet cells, seen with the absence of green staining. When switched into our differentiation medium, the bottom left-hand image shows the cell proliferation decreases but doesn't completely abate, and differentiation occurs, again, denoted by green as the MUC2 positive goblet cells, but also these small pockets of yellow staining that you can see, which is that small proliferative niche that I mentioned. On the top right-hand side, you can see an absence of enteroendocrine cells and enterocytes in the proliferative state, and an increase in both of these cell populations once switched into differentiation medium, as seen by the presence of green enteroendocrine cells and yellow enterocytes. I noted that proliferation isn't completely abated, and this is due to the retention of a small LGR5 positive cell population within the cultures. And this is important physiologically, as the intestinal lining in vivo is a place of flux with continuous renewal of aging cell populations to maintain barrier integrity. This cell proliferation and replacement is mimicked in vitro by this small LGR5 positive cell population, and enables these cultures to be maintained for up to 2 weeks. So now that we have reviewed the basic biology of these systems, I want to switch gears and focus on how intestinal organoids can add value to drug discovery endeavors. However, before giving you info about the applications of these organoids, we'd like to know if you use organoids in your research. Again, I'll give you a second or two to vote. As you can see from the table on the right, gastrointestinal side effects are the most common location of adverse events in patients enrolled in clinical trials. This is perhaps not surprising given the gastrointestinal tract is the first point of contact for drugs entering the body via oral administration. This is also the site where these drugs are initially absorbed and metabolized. A healthy, intact intestinal epithelial lining plays a key role in modulating immune responses via hormone secretions and chemical crosstalk. Disruption of the barrier this lining provides is often manifested in symptoms such as diarrhea, dehydration, and ulceration, which increases the susceptibility of the patient to infection. Therefore, any drugs that affect the cell viability, proliferation, or integrity of the barrier itself contribute to toxicity. That is often the limiting factor for novel therapeutic drugs, and can also be a dose-limiting factor for the efficacy of the drug in the clinic. The classical drug discovery paradigm involves cell lines and animal models. In the intestinal sphere, those cell lines are often Keiko2 cells. While these cells have been favored by researchers for a significant amount of time due to their hardiness and ease of expansion, it is also simultaneously recognized that these cells are immortalized and lack the secretory cell phenotypes found in vivo. In addition, due to the cancerous background of the cells, they also lack key pathways, receptors, and transporters you would find in the cells of the intestinal tract in vivo. Animal models have the advantage of being more biologically complex than cell lines. However, there is often a lack of translation of animal data into humans, most likely due to the difference in the biology between species. Also of note is that animal models, even rodents, are not amenable to high throughput screens. So when looking at multiple compounds, there is a trade-off between throughput and staying close to the biology in question, so as to make the data as informative as possible. And this is where the potential for human intestinal organoids lie, to provide that bridge in scalability while staying close to the biology. The use of human intestinal organoids enables researchers to create biobanks that have diverse donor backgrounds from both healthy and diseased tissues. Human intestinal organoids have been used recently as a personalized medicine approach via a collaboration between the Hub and Vertex Pharmaceuticals, where they used rectal tissue from cystic fibrosis patients to screen for the efficacy of drug combinations on those patients. With our ability to standardize media formulations to both expand and differentiate human intestinal organoids and keep these organoids in culture long-term, our intent is to provide screening services for our customers, which generates informative data for their projects. There are a variety of different technologies at various stages of development on the market, from co-culture models, organ on a chip, and microphysiological systems. We believe that offering human organoid services is the first step to discerning what the predictive potential of these systems could be for our clients, so that decisions can be made on their end as to how and when they adopt these technologies as part of their internal drug discovery processes. And with that, I'm going to hand over to Danny, who will talk about the services we have currently launched and the data to support the inclusion of these services into your early drug discovery due diligence. But first, another poll. What kinds of applications are you planning on using organoids for? I'll give you a second or two to vote. Okay, now it's over to Danny as promised.
I'd like to take this opportunity to provide some technical information about how we've developed the workflow and the tools we use to leverage organoid technology as a model for intestinal physiology and assessing potential drug toxicity. We've established organoid lines directly from patient tissues derived from the duodenum, ileum, and colon regions using stem cells in testicle media. Intestinal organoids are expanded in intestinal organoid growth medium, or OGM, and seeded into 96-well plates in matrigel domes. Cultures are allowed to establish for 2 days, then treated with desired drugs for up to 5 days of continuous exposure, with media and drugs replenished every 2 or 3 days or as needed. We've used this workflow to test a number of compounds that have known intestinal toxicity, such as gefitinib, colchicine, bifluorouracil, and flavopiridol. These compounds can act through various anti-proliferative mechanisms that inhibit growth as well as display cytotoxic activity. Here we have some representative data where intestinal organoids derived from various regions, as well as distinct individual donors, were cultured in intesticle OGM and are treated with 5-fluorouracil or flavopiridol. 2 well-studied compounds expected to be toxic to proliferating cells. Organoids were established as described in a 96-well plate format and treated with escalating concentrations of 5-FU or flavopiridol. And a dose-dependent inhibition of organoid growth can be observed, as well as decreased cell viability, which is plotted in these graphs. The IC50 concentrations were interpolated. In addition to our intestinal lines, dose response curves are also generated for 2 intestinal immortalized cell lines, the KCO2 and the HT29 cells. While there is donor-to-donor variability amongst the primary organoid samples is observed, the confidence interval for the organoid lines is quite tight, and the average IC50 across the organoid donors was lower than the IC50 values generated using the KCO2 and HT29 cell lines, demonstrating the value of organoids as sensitive models for drug testing. IC50 values in the low to sub-micromolar range were expected for these compounds, and that is the result that was observed, demonstrating that the organoids reliably detected the expected toxicity of 5-FU and flavopiridol. To demonstrate assay-to-assay reproducibility and interoperator variability, we use duodenum, ileum and colon organoid lines derived from a single healthy donor to profile the effect of gefitinib and colchicine on organoid viability over multiple and independent experiments. One can see from these results that the variance of the dose response and the IC50s to be quite tight throughout independent runs. The CACO2 line was also characterized and again demonstrated much lower sensitivity to gefitinib and colchicine, indicating that CACO2 would be a less predictive model for GI tox. Also, which might be of interest, are some microscopy images of the organoids, which, though non-quantitative, clearly capture the dose-dependent effect on the morphology and overall condition of the organoids. In addition to using organoids as a predictive model for gastrointestinal toxicity, I'd like to spend some time discussing the use of organoids to model barrier function of the intestinal epithelium. The intestinal epithelium maintains barrier integrity through a complex series of tight junctions in between cells. Of course, the barrier is not some impermeable or impenetrable structure, but rather has selective permeability to things like vital nutrients and smaller molecules that may diffuse or be transported across the barrier, while large molecules or bacteria of the microbiome remain in the intestinal lumen. The barrier is maintained by complex structural and molecular interactions, such as the mucosal layer, the tight junctions, and continuous renewal of the epithelium in differentiated cell types from a pool of pluripotent stem cells that reside in the intestinal crypts. Disruption or loss of barrier function can carry significant implications and outcomes such as inflammation, dysbiosis, and sepsis. And indeed, intestinal barrier dysfunction has been associated with various diseases, as well as adverse clinical outcomes as a result of drug-induced gastrointestinal toxicity. One commonly used model is the well-known CACO2 that we've discussed previously. And in the following slides, we'll have some data exploring the use of the organoid technology to model barrier function and how it compares to the CACO2 line. In the barrier model, we can culture and expand the organoids in 3D matrix culture, exactly as previously described. To establish the barrier model, the organoids are harvested, dissociated into single cells, and then seeded onto permeable membrane supports, such as the transwell system, which are well known in this type of application, and grown to form a complement monolayer. Once seeded into the transwells, the organoids are maintained in intestinal differentiation medium, or ODM, which promotes the differentiation into more of the intestinal specific cell types, rather than the higher proliferative culture that the OGM supports. With ODM cultured monolayers, we see an increase in markers typically associated with these differentiated cell types, both through gene expression and immunohistochemistry. Once the monolayers have been established, they can either be maintained in this, quote, submerged state, or they may be further differentiated into an air-liquid interface, or the ALI culture, by removing the media from the apical compartment for an additional culture period. Once seeded, differentiation in the submerged state takes roughly 1 week, while the ALI culture is an additional 7 to 10 days of culture. Comparatively, the CACO2 line is typically cultured for a total of 19 to 21 days. The ALI culture results in the formation of the mucosal layer and higher expression of certain functional protein families, such as transporters and metabolic enzymes. However, both the submerged and the ALI cultures form intact barriers, and data in the following slides were generated using the submerged procedure. One method of assessing barrier integrity is through the measurement of the transepithelial electrical resistance, or TEER value. Many of you are undoubtedly familiar with the transwell system, which are a supported insert that sits inside the well of a culture plate, while a 2-arm probe is used to position electrodes on either side of the monolayer to measure the electrical resistance across the membrane. Higher resistant values imply a tighter barrier and cell-cell junctions. Disruption of these interactions would result in lower resistance values, as would any wells that were non-confluent or insufficiently differentiated. In this figure, we've cultured organoid lines derived from different donors and regions and compared the TEER values between the ALI and submerged cultures, as well as to the commonly used CACO2 line. We can see that there is donor-to-donor differences, as well as region-to-region differences. And notably, there is an overall increased TEER value of the organoid line compared to the CACO2 line. While TEER is one method to assess an intact barrier, one can also use the permeability of a reference or marker molecule with known low permeability characteristics to assess the barrier. In this scenario, once the monolayer has been differentiated, the marker molecule can be added to the apical compartment, and its net flux across the monolayer is quantified over a specified time period. Any analyte with low permeability can be utilized in this manner, so long as you have a method of quantification. Of course, there are also practical concerns, such as the limits of detection, throughput of the actual detection method, and whether that marker molecule is potentially metabolically labile or transporter-mediated. In this example, we've selected a FITC labeled large molecular weight dextran as a low permeability marker. Its 4,000 to 5,000 Dalton size and high hydrophilicity renders the rate of permeability across an intact monolayer to be quite low. In comparing the CACO2 monolayer with a colon-derived organoid monolayer, we can see that the net accumulation of the FITC dextran is higher in the CACO2, suggesting a leakier barrier. When coupled with a compound known to have disruptive effects, such a marker molecule can be used to quantify those effects. In this figure, we have a dose-response curve of colchicine against organoid monolayers derived from multiple intestinal regions, as well as the CACO2. We used endpoint tear as well as accumulation of the FITC dextran in the basal compartment to assess the effect of colchicine and demonstrate here that the decrease in tear values are accompanied by an increase in the net transit of FITC dextran across that monolayer. In contrast, there is no effect of the CACO2 over the same concentration range, demonstrating the greater sensitivity of the organoid lines to this compound with known GI toxicity. STEMCELL recently launched a service based around the barrier function utilizing organoid-derived monolayers. In a modification of the procedures used in the previously shown data, the standard service has been adapted to a 96-well plate format for higher throughput and incorporates 3 readouts that are generated from the same assay wells. First is the 4K FITC dextran that is added alongside the compound being tested and is present during the full duration of the compound treatment. At the conclusion of the compound exposure, there is a washout followed by an endpoint permeability assay using the small molecular weight, low permeability marker lucifer yellow. By using lucifer yellow, at the very end of the assay, we can take a quote snapshot of the barrier state after any compound treatment. And then finally, the viability is assessed using cell titer glow. When re-examining the effect of colchicine using the standard service format, again, we can clearly see the dose response in a colon-derived organoid monolayer, but not the CACO2. These effects are readily apparent in the FITC dextran readout on the left, the lucifer yellow in the center, as well as the cell viability readout on the right. A similar result is observed with sapitinib, which is an EGF receptor antagonist, a compound also with known adverse GI toxicity. Again, we see an increase of the FITC dextran and lucifer yellow permeability with a corresponding loss of cell viability. In contrast, the CACO2 line does not have a similar response and would thus be a less predictive model for both of these compounds. In summary, STEMCELL has developed organoid-derived systems that more accurately model intestinal physiology that can capture responses that are not observed with commonly used immortalized cell lines. By using these systems in assay-based applications, we can provide new relevant tools to evaluate compounds for gastrointestinal toxicity and effect on barrier integrity. Now, I'd like to take a brief moment to talk about a similar service STEMCELL has been offering that utilizes mouse organoids instead of human organoids. The workflow is very much the same as human in terms of organoid culturing and compound treatment schemes, though the media composition is different. However, STEMCELL has developed a proprietary image-based analysis algorithm that can quantify individual organoid morphology. In this assay, high-resolution microscopy images are taken of each well in 3 separate focal planes and then flattened or composited into one image. A set of images is taken just prior to the compound treatment to establish a baseline state of morphology, and then another set of images is taken at the endpoint of the compound treatment. The image analysis algorithm is then used to identify each individual organoid object and its properties, such as size or cross-sectional area, and branching or bud score is determined. Once the images are taken, the viability can then be assessed with a reagent-based assay such as the cell titer glow again. Here we have a dose response of flavopiridol analyzed using the image algorithm. While the effect on organoid health in terms of size and branching compared to the solvent control is readily visually apparent, it's impossible to quantify those effects without the algorithm. Even this small study alone was comprised of over 7,000 individual organoid objects that the algorithm identified and scored to make this figure. It's a very powerful tool for those who may be interested in morphological readouts. And thank you so much for your attention.
Thanks, Danny. So that was a lot of information that has been provided in our presentation today. Before we wrap up here, I want to put a final poll out there. Which organoid tissues would you like to see explored next? Again, I'll give you a second or two to vote. So I want to end this webinar by taking a step back from intestinal organoids and providing some information about contract services in general here at STEMCELL Technologies. One of the benefits of working with our team that I want to highlight is the expertise that spans the media products themselves and the commercialized assays that incorporate these products. We are ISO 13485, 9001, and 1401 certified, and we have been around a long time, over 20 years and counting. So we have a wealth of expertise in a variety of areas, and we are more than happy to answer any question you may have about your service needs. Our most popular standard services are highlighted here. They are grouped into hematopoietic, immune, mesenchymal, and intestinal. The latter is what we've covered today. Should you have any questions on how these services could be combined or be used as a standalone service, feel free to use the website listed here for further information. Our most popular services are our hematopoietic CFC and hematox assays. The CFC assay in particular has a wide range of utility. Not only is it used to discern the toxicity of drug candidates on the hematopoietic stem and progenitor cell populations, but it is also being used by researchers in the gene therapy space who are using gene-edited CD34 positive cells and those in the immuno-oncology field studying CAR-T therapies for AML patients. We can also support non-standard studies that require contract assay development. However, given the bespoke nature of these types of services, I would encourage you to reach out to us directly should you have any questions related to contract assay development. We do have 2 new services coming online in the near future using our human intestinal organoids. The first is our human intestinal absorption assay using a similar assay format to the barrier integrity assay that Danny covered today. This assay can evaluate how your novel compound is metabolized when taken up by the intestinal epithelium. We also have an efficacy service launching aimed at researchers looking into therapeutics for inflammatory bowel disease, specifically Crohn's disease. The goal of this assay is to screen for the efficacy of various Crohn's disease drug candidates to reduce the leakiness of the intestinal barrier by assessing the paracellular permeability of tracer reagents. If you have any questions about these or any other services that have been highlighted here today, please take a moment to click on the QR code in the bottom left corner, which will link to our contact information. And I'll give you a second for those that want to take a quick snapshot of that QR code. So, in conclusion, the message I hope you're taking away with you today is an understanding of the human intestinal organoids grown in stem cell media and used in our services, and the potential advantages of these organoids compared to other research tools, the flexibility of our culturing systems and the assays they enable, and how STEMCELL consistently strives to embody our motto of scientists helping scientists by providing products and services to meet the needs of our global customers. I would like to highlight and acknowledge the many stakeholders involved in producing the webinar we have brought to you today. Our scientific contributors are Wyatt Anderson, Jill Brandon, Kathy C., Martin Stahl, and Ryan Conder. Our product management contributors are Alexander Schlatt and Valerio Russo. Our program manager contributor is Johnny Ather. And finally, our executives, our head of R&D, Sharon Lewis, and our owner and CEO, Alan Eves. These services are provided to our customers due to a license agreement with the Hub. Researchers who avail themselves of our human organoid services do not need any additional licenses from the Hub in order to use the data we generate on their behalf. STEMCELL is a global company and our services are global. With that said, our headquarters and service labs are based in Vancouver, beautiful British Columbia, Canada. STEMCELL acknowledges that our work spans many territories and that our head office is located on the traditional unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. Finally, should you have any questions after this webinar, please feel free to contact us directly or contact your local STEMCELL sales representative. Thanks again for joining us today. It was a pleasure to be able to showcase our efforts in organoid services, and we will be happy to answer any questions at this point.
Fantastic. Well, thanks so much, Victoria and Danny, for the great presentations today. With that, we'll move right on to the Q&A. So I'll invite Victoria and Danny back onto the line here. All set to answer a few questions, Victoria and Danny?
Yes, absolutely.
Awesome. So first question here, how did you measure the cell viability of the organoids?
Sure, I can take that one. Thank you for the question. We actually had that question from multiple participants. So typically we use Promega's cell titer glow. When the organoids are cultured in the suspension matrigel format, we use the cell titer glow 3D, which is optimized to liberate cells from extracellular matrices. For the monolayer barrier assay, it's just the traditional or the normal Promega cell titer glow. And presumably these organoids can also be measured the viability through other common viability assays, but we've standardized our procedure and our assay workflow with the Promega kits.
Fantastic. Next question here, what types of characterizations can be done with some of the newer assays that you mentioned towards the end? And are there any potential add-ons?
Oh, sure, thank you. I can address that one as well. So we weren't able to show all of the data that in the time allotted, but we've started to characterize these organoids for some of the physiological characteristics, such as transporter expression and function, as well as cytochrome P450 metabolism. And we've demonstrated internally that the organoids have a much higher level of transporter expression from a functional standpoint, looking at known transporter substrates and measuring the flux across, like in the monolayer format, where we can use the monolayers in a permeability assay and measure the flux in both the apical to the basal and then back from the basal to the apical and calculate the efflux ratio to demonstrate functionality. And also we can use known transporter inhibitors to basically block that efflux. And when we compare with say the KCO2, which is very commonly used in the permeability assay, that there is significantly higher transporter function in our organoid monolayers, which is also region dependent. And the same also goes for the cytochrome P450 activity. The KCO2s are famously low for metabolizing enzymes. And the organoid models that we've been working with have demonstrated significant, orders of magnitude higher metabolism. As far as potential add-ons for these assays, while we don't offer it as a standard part of the service, some target based characterization like qPCR expression or even immunohistochemistry is possible. Thanks for that question.
Excellent. Yeah, thanks for the answer. All right, why did you put the organoids in suspension 7 days before applying them to the monolayer culture?
I can answer that one as well. Thank you. So as Victoria mentioned, we currently have two media types. One is the organoid growth media and the organoid differentiation media. The growth media maintains them in a more proliferative state, and we use that suspension to basically, as part of the expansion phase, to basically grow enough organoids to be able to apply to our downstream readouts, like for instance, like the barrier. So the suspension phase beforehand is essentially an expansion. And then once they're seeded, we basically seed them close to confluency. And then when they reach confluency is when you'll really get that differentiation into all the various cell types that would make up a monolayer. Thank you.
Excellent. Have you applied the image analysis algorithm to the human PDOs?
Oh, yeah, that's a great question. We actually had that from multiple participants today as well. The short answer is it's something that we're working on, and image-based analysis is something that we are developing for the human organoids. One of the I don't want to say limitations, but one of the hurdles that we have to overcome is that if you recall some of the earlier microscopy images that we showed of the proliferating human organoids, they tend to get quite large and they have a cystic morphology, but more importantly, they tend to sort of overlap in that 3D culture. So a microscopy image is obviously, it's a 2D image of like a 3D culture. And if you get organoids growing in, say, different planes, but overlapping, then when you look at it from like a cross-sectional area, it's very difficult for algorithms to resolve objects that are overlapping. So part of the optimization that we did for the mouse one is the seeding density has to be high enough to give you enough organoids to be able to run your analysis on, but also not so high that they overlap and it confounds the algorithm. So we are making advancements in human organoid image-based analysis, and it's definitely like a goal, but we aren't quite there yet. Right. Thank you for the question.
Fantastic. So how do you discriminate the remnant FITC exposed along with the experiment from the end readout, the Lucifer yellow, considering the emission spectrum?
Oh, yes, thank you. Yes, so you're right that, so the emission spectrum from those two fluorophores do overlap, even though the excitation is quite different. So we can't apply the FITC and dextran Lucifer yellow concurrently. So how it's done now is that the FITC dextran is added at the beginning of the compound treatment, allowed to accumulate during the drug exposure, and at the end of the drug exposure, we do a washout of the drug as well as the FITC before we add the Lucifer yellow. So they're not there at the same time. And in a related sort of aspect, we've also considered looking at the large molecular weight dextrans that may have other fluorophores attached besides FITC, and they are available, and they're also available in different molecular weights. So if there's an interest in looking at both a large and small molecular weight in the same readout, like concurrently, then it may be possible to do it with a different fluorophore marked dextran, or perhaps instead of the small molecular weight Lucifer yellow, find another molecule that would not interfere in terms of excitation and emission. I would also add that as part of our characterization of these organoids, we also do a lot of LCMS, so like mass spec, and of course that opens up all sorts of possibilities in terms of what sort of molecules that you might want to measure going across like that barrier. For the purpose of the service, for logistics and ease of use and throughput, we selected these fluorescent molecules. But if there's an interest in looking at, say, small molecules in which traditional LCMS may be required, that is certainly a possibility. Thanks.
Excellent. Yeah, thanks, Danny. All right, can you expand on the organoid analysis software? Can it be used for human organoids? Does it need a specific microscope, and is the public able to access it?
Oh, right. So the analysis software is something that was proprietary and developed internally here at STEMCELL. As for the human organoids, that we talked about earlier, there are some challenges with, theoretically, it can be used, but there are some challenges just based on the human organoid morphology. We're currently using a Zeiss AxioVert microscope, where we can basically take cross-sectional images at different focal planes, and presumably any microscope that has that capability can do that. The reason why we take the 3 planes is because the Matrigel dome is like it's a structure that has like depth, and the organoids can grow anywhere in that plane. And by taking multiple focal planes, we can ensure that at least every organoid is in focus in at least one of those planes. And it's through optimization that we arrived at like three planes. Presumably, if you had a very small dome, you might be able to get away with one, for instance. And as far as the access, like the mouse organoid assay, we have as a standard service now. So if somebody was interested in doing this sort of morphological readout, it is a service that we do offer for the mouse organoid. Thank you.
Perfect. How customizable are the assay services assays that you mentioned?
Oh, thank you. In terms of customizability from, I guess, like from an end user standpoint, so we don't, stem cell services are, like, we tend to construct them like on demand and as per the need like of the customer. So like we don't require like like a specific number of compounds or only certain number of concentrations. So all those are all customizable. Things like treatment times, like how long you want your compound exposed for, that's also for the most part, open for whatever the customer needs. Of course, there are limitations as to how long we can maintain, for instance, like a 3D organoid, in culture before they start to overgrow and are no longer viable. But ultimately, we would work with you individually to basically customize each assay to your needs.
Perfect. No, it's great to hear that stem cells so flexible. All right. Next question here. Do barrier integrity effects tend to be more or less sensitive endpoints compared to cytotoxicity measures? And is that consistent between the CACO2 and organoid models?
Thank you for that question. So if you look at some of the data that I had around for the barrier readout, typically we would see an effect on the barrier integrity as measured by the flux of, say, one of our marker molecules, like for instance, like the phytidextrin. You'll see the effect in the flux of that molecule before you'll see a loss of cell viability. And then is it consistent between the CACO and the organoid models? Yes, in that you'll see an increase of the marker molecule flux before you see an impact on the overall cell viability. But it's also compound dependent that you may see that effect at much lower concentrations in the organoid models, like versus the CACO models. And for some molecules, they're actually quite similar between the two cell lines. But the basic answer would be that you would see the barrier integrity effects at concentrations lower than you would see the viability effect.
And I just want to add here as well, as that's quite important from an in vivo aspect when you're looking at potential drug candidates that may increase the leakiness of the barrier, which would then elicit an immune response from the patient itself, without that loss of barrier tightness being related to cell death. So it's the reason why we have the two services there is some people may be looking at how toxic their compounds are to the actual cells themselves, but others may be interested to know whether you have changes in the tightness of the barrier along the gastrointestinal tract that aren't related to autophagy of the cells themselves or cell death in any way. And like I say, because certain areas of the intestine are quite tightly linked for reasons of bacterial presence in the lumen. Then that's why these two services and the difference you can see between an effect on barrier integrity prior and preceding to cell death are important for some of our clients.
Perfect. Yeah, great answer, Victoria and Danny. All right, can you please comment on the organoid technology that's available? You know, part of the organoid technology, like supplements and media, seem to be patented. So how does this affect your services?
Oh, I can take this one. So I would just say, I think I touched on it towards the end of the presentation. So we have a partnership with the Hub that allows us to offer these services via the patents that are part of their technology. So unlike media, organoid media, where you would need licenses to use the technology in-house, you don't need to pay any additional licensing fees, and there's no barriers to freedom to operate of the data that we generate. So we have the ability to offer the services to you to generate the data, and you can use the data however you wish in your research without needing any other licenses.
Perfect. All right, this question was asked sort of towards the start of the presentation, but if growth is subdued, then how will the body repair or doesn't it at this stage?
I think, again, that comes down to, you know, when you have that delay, I think, and I'm just going off the top of my head here, that the renewal of the cryptovillus access is around every 3 to 4 days. So if you have a delay in that due to unexpected cell loss, where you've got gaps in the barrier, or you've got a slowdown in the ability of the cells to proliferate, then you are creating that breach of the barrier that could create an immune response that exacerbates that wound healing. So anytime you've got pro-inflammatory cytokines in the mix, you're going to have further knock-on effects of the epithelial cells of the lining themselves. So, you know, in that respect, that lack of ability, or that unexpected loss, I would say, of cells can lead to ulcerations in the region themselves that then become self-perpetuating as an immune response. Sometimes hinders the healing. So it can be quite dramatic by just having a small increase in cell viability in those particular regions themselves. And depending, I would say as well, as, and this is maybe going into some of the IBD stuff, depending on whether you have any form of intestinal diseases, that could be even more drastic. We don't have any data to support that yet, but it is something that we're looking at.
Excellent. Thanks, Victoria. All right. So how far in advance are they to be taken before the drugs are administered? Sorry, I'm not really totally sure what this was about, but it was asked again towards the start of the webinar here.
Sorry, I don't understand the question.
Yeah, it's hard to say. It was asked towards the start, so maybe
Danny, to maybe how long we culture the cells prior to seeding them down. Is that maybe the question? How long we have to culture them to get enough biological material?
Yeah, that very well could be.
Perhaps I could just maybe just describe a bit of the work, like very briefly, the workflow that may answer this question. In our tox assay, that since we maintain them in our organoid growth medium in the proliferative state, it can basically be assayed when there's enough material, like expanded enough to accommodate the entire study. For the barrier assay, because the monolayer has to form, the tight junctions have to form, and differentiation has to occur, we find that we need to maintain that monolayer from seeding for at least, we standardize around 7 days, but probably like at least 5 or 6 days minimum before we would do any drug testing. But also once they're differentiated, they can be maintained in that monolayer state for like quite a while. But I would say probably for the monolayer, at least a week prior to any drug administration.
Yeah, perfect. Thanks a lot. All right. Do you offer media for breast cancer organoids?
That is a good question. I believe we definitely have products to support breast cancer. I am unsure whether those are media or accessory products. The advice that I would give there is to head onto our website, stemcell.com. And our pages are quite easy from a product standpoint to navigate based on the tissue of interest.
Excellent. All right, next one. For HTS, how do you get a smaller CV percent and higher Z when using organoids?
I think that comes down to the techniques themselves and some, I would say, struggling to answer this one. We do use a certain technology in-house that hasn't been launched yet as a commercial product that we feel adds a lot of value with regards to reproducibility based on the readouts that we're using. So we can't answer that fully yet because it isn't a launched product. The product itself is a type of plasticware and it will be released soon, but we can't say anything at the minute because it's not released. I would also say as well as it is part of developing the assay itself that there's a certain amount of familiarity that our operators need in order to be able to decrease the amount of variability both within a plate and also between the plates. And it's why we spend an extensive amount of time during the commercialization phase of our products into services by ensuring that the operator variabilities are within our own parameters for whether we class something as a robust assay that's amenable to 96 and 384 well formats.
Right. Yeah, that makes a lot of sense. Thanks, Victoria. All right. So to study intestinal organoid metabolism, do we need to culture the organoids in a growth media or a differentiation media?
Right, I can take that one. So for the characterizations that I've done around the cytochrome P450 metabolism, the differentiation media definitely promotes higher cytochrome P450 activity. And I've been doing quite a bit of work recently in our monolayer culture for metabolism. And not only that, we find that the ALI culture actually also has higher metabolic capability than the submerged culture.
Excellent. Thanks, Danny. How do you transform the intestinal organoids in suspension prior to seeding as monolayers?
Oh, right. Yes. So during the expansion phase, we will grow them in 3D culture in a matrix gel extracellular matrix, and then we will essentially single cell them, or basically disrupt them into, well, as close to single cells as possible for seeding onto the monolayer. And that's basically it's a combined technique with basically like a trypsinization as well as mechanical disruption. And it is one of the factors that goes into creating like a good monolayer, is having a good, if not single cells, as close to possible prior to that seeding. Thanks.
Perfect. Now, this question is going back to the last one, but this person is asking how long we can culture organoids in the differentiation media. They said that they need to study Paneth cells for a week. Would that be possible?
Yes, that's certainly possible. Once they're differentiated, they can be maintained for like quite a while. And while I haven't done it specifically because it's not usually in our study design, our R&D group has maintained like monolayers for on the order of weeks. And then once they're differentiated, all the physiological characteristics and gene expression should be quite stable. Is what I'm believe in discussion with our R&D group.
Excellent, thanks, Danny. Have you tried to use gut epithelial cells derived from diseased patients and healthy donors, and do you see any differences differences between the two. So they say, their question aims to see whether in your organoid culture media you observe the pathogenic phenotype, like inflammatory cytokine secretion, from the diseased organoids other than the healthy organoids.
Do you want me to take this, Danny, or do you want to take it?
Yes, perhaps you could address this one.
So we're currently looking at diseased patients right now. We have a Crohn's disease patient that is making up our IBD model, and the plan is to do some characterization of those disease cells compared to the donor banks that we have of healthy tissue, and see where we have maybe differences in gene expression or protein expression, and whether we have any differences around functionality of surface receptors, and whether we have any difference in readouts with known drug compounds, but we don't have any data to share right now or anything further to say at this point.
Excellent. So what is the average size of compounds that people are giving to you right now, and how many can be screened?
Oh, sure, I can address that one. When you say size, if you mean like the size of the study?
Yeah, the volume, I think, of compounds.
Oh, as opposed to like the molecular weight. Okay. Yeah, in terms of, it's very common that we see studies in the size of 5 to a dozen compounds, 8 to 10, with dose responses between 6 and 8 concentrations, done in triplicate against multiple lines. So if you do the math, there is probably at least several hundred data points. In one study, we can probably accommodate well over 1,000, maybe somewhere around 1,500 like data points in one comfortable study. So if you do the math, backtrack the math there, you could probably do something on the order of accommodate easily maybe 20 plus compounds, full dose response of eight concentrations in triplicate, and that would be well accommodated with our workflow.
Perfect, thanks a lot. And I think in the interest of time, we'll just ask one last question here. Does the oxygen concentration have an effect on the ALI organoid culture?
Yes, I can address that one. So yes, oxygen is definitely one of the driving forces, part of that ALI differentiation. And as you get more oxygen, it does help drive that differentiation. And then while in the submerged state, it drives more of an undifferentiated wound repair mechanism. So while we don't really characterize, like in our assay, I haven't really characterized specifically like how much response you might get at different oxygen concentrations. All of our assays are done just at ambient oxygen concentration, but presumably, if you were to say run it in like not an anaerobic but like a hypoxic condition, then that differentiation would likely be affected.
I will just add, it's not our own data, it's data from Stappenbach, who's a KOL in the field. He has some nice convincing data that oxygen is one of the driving forces between wound repair and differentiated phenotypes in the monolayers. So the ALI definitely does get more oxygen just because of that surface being exposed. And that does help drive differentiation. And when submerged, it drives an undifferentiated, more wound repair phenomenon. So you can use both systems, both submerged and the ALI, depending on what you're trying to discern, whether it's cells in a more differentiated state or whether it's more of the expansive phase undifferentiated wound repair style mechanisms.
Perfect. Yeah, thanks both. And maybe we'll just squeeze one last question in. I think it's a good one. Do you have any data for transporters like PGP or BCRP, as well as intestinal mechanism like CYP3A4 and UGT?
Right. Yes. So I've been doing some characterization recently in intestinal monolayer, and yes, we have some very convincing data that there's absolutely fairly high levels of PGP expression, and there's I also have some preliminary data around the BCRP transporter, and those are both done looking at the efflux of marker substrates. Both in the apical to basal and the basal to apical, and then calculating the efflux ratio, but then also following that up with inhibition with known transporter inhibitors to basically block that efflux ratio. Sorry, what was the second part of that question?
So any data for transporters as well as intestinal metabolism like CYP3A and UGT?
Oh, right. Yes. So we have done some characterization around the cytochrome P450s, and we have shown that there is definitely like CYP3A4, 2C9, and 2D6 activity. The UGTs and the other phase 2 metabolic enzymes, I have not yet characterized.
Excellent. Well, Danny and Victoria, thanks so much for all the great insights today, both in your presentations as well as the Q&A. It's really been a pleasure having you with us.
Thank you, and thanks for all the great questions.
Yes, thank you so much. This has been really informative.
Yeah, definitely. A big thanks to the audience for participating. And in closing, we hope you enjoyed the Scientist.com and STEMCELL Technologies webinar, and we'll see you again next time. Have a great day, everyone.